Welcome plus 2 first year science student. Last class we discussed about the structure and function of nucleic acid. That means structure and function of DNA and structure and function of RNA. Today's topic is carbohydrate and amino acid. I shall to discuss about carbohydrate and amino acid. We know carbohydrate and amino acid are the biomolecule which present inside the living cell. Carbohydrate are the macromolecule, whereas amino acid is the micromolecule. You know macromolecule and micromolecule, there is some distinction between macromolecule and micromolecule. Macromolecule are those molecules whose molecular weight is about more than 1000 Dalton molecular weight, but micromolecule are those molecules whose molecular weight is less than 100 Dalton. You can distinguish between micromolecule and a macromolecule, but here carbohydrate is the macromolecule and amino acid is the micromolecule. Then comes to the carbohydrate. We know carbohydrate is the photosynthetic capital of the plant. Why it is considered as a photosynthetic capital of plant? Because carbohydrate forms about 80% is the important constituent of the plant body. So carbohydrate is the important constituent of plant body. So this is also called as photosynthetic capital of plant. But we know about 1 to 2% carbohydrate are the constituent of the animal body. We know also carbohydrate are the fuel molecule that generates ATP during cellular respiration. We know carbohydrate are the substrate molecule that oxidize during the process of respiration that gives energy. We know 1 mole of glucose oxidized to produced in 36 molecule of ATP in presence of oxygen. So carbohydrate are considered as the fuel molecule that generate ATP on combustion. Similarly, we can say carbohydrate is a organic macromolecule, that is the polyhydroxyaldehyde or polyhydroxyketone and their derivative. Means it is a macromolecule which contains many alcoholic group as well as aldehyde group and ketone group and their derivatives. So carbohydrate are the photosynthetic capital of the plant and also fuel molecule that generates ATP during complete oxidation. Then we can say, what is carbohydrate? Carbohydrate, hydrate of carbon. Hydrate of carbon means that is water of carbon. That means carbon water. Here we can say that hydrates of carbon. Why it is considered as hydrate of carbon? That is H2O carbon, means carbohydrates of carbon. That means it can be considered as CnH2ON. Here the number of water molecule is that is considered as N, the number of carbon atom that is N. So its general formula is CnH2ON. Means we can say that CnH2n ON. It can be also written as CnH2nON. So this is the general formula of the carbohydrate. We cannot say the general formula of carbohydrate, but this is the general formula of monosaccharide. Okay. These are the introduction of the carbohydrate. Then comes to the classification of carbohydrate. Generally, carbohydrate classified into 4 major groups on the basis of presence of number of sugar unit. First one is monosaccharide, second is disaccharide, third is oligosaccharide, fourth is polysaccharide. Monosaccharide means what? Mono means single, saccharides means sugar. Single sugar unit present in those carbohydrate, that is called as monosaccharide. Then second is disaccharide, when two sugar residues are combined with each other forming disaccharide. Similarly, oligosaccharide means when 3 to 7 sugar residue combined with glycosidic bond that form oligosaccharide. Okay. First one is monosaccharide, then disaccharide, then third number is oligosaccharide, fourth number is polysaccharide. But here monosaccharide means single sugar unit present in those carbohydrate, that is called as monosaccharide. Similarly, when two sugar residue are combined with glycosidic bond, that form disaccharide. Similarly, when 3 to 10 sugar residue combined by glycosidic bond, that form oligosaccharide. Okay. Then polysaccharide, when more than 10 monosaccharide unit, more than 10 sugar residue combined by glycosidic bond, that form polysaccharide. Okay. Then comes to first one is monosaccharide. Monosaccharide is also called as simple sugar. Why it is considered as simple sugar? Because it cannot be hydrolyzed into further sugar unit. So this is called as simple sugar. It contains 3 to 7 number of carbon atoms. Monosaccharide are the monomer of the carbohydrate. Then comes to the monosaccharide cannot be hydrolyzed into simple sugar. On the basis of presence of carbonyl group, monosaccharide is of two types. Carbonyl group means aldehyde group and ketone group. If the monosaccharide contains aldehyde group, that is called as aldose sugar. If the monosaccharide contains ketone group, that is called as ketose sugar. Suppose aldehyde group is CHO. Similarly, ketone group, that is C double bond O. Here, if the ketone group is present, the monosaccharide is considered as a if the ketone group is present, the monosaccharide is considered as a ketose sugar. Similarly, if the monosaccharide contains aldehyde group, that is called as a aldose sugar. So all monosaccharides are classified into two types. One is aldose sugar, second is ketose sugar. When aldehyde group is present, that is called as a aldose sugar. When ketone group is present, that is called as ketose sugar. That is ketone group, C double bond O, that is ketone group. Similarly, CHO, that is called as aldehyde group. When aldehyde group is present, that is called as aldose group. When ketone group is present, that is called as ketose sugar. Here monosaccharide can be classified into different groups according to their presence of number of carbon atoms. That may be, name of monosaccharide may be triose sugar, tetra sugar, pentose sugar, hexose sugar, heptose sugar. Here 3 to 7 number of carbon atom present in the monosaccharide that can be classified into 5 groups: triose, tetrose, pentose, hexose, heptose. When the number of carbon atom is 3, the sugar is called as triose sugar. Its molecular formula is C3H6O3, and aldose form of triose sugar is glyceraldehyde. Similarly, ketose form of triose sugar, that is dihydroxyacetone. Then when 4 number of carbon atom is present, the monosaccharide is called as tetrose sugar. Its molecular formula is carbon 4, hydrogen 8, oxygen 4. Aldose form is erythrose and threose, but ketose form of tetrose is erythrulose. Then comes to when the 5 number of carbon atom, the number of carbon atom is 5, the sugar is said to be as pentose sugar, or 5 carbon containing sugar, that is called as pentose sugar. Its molecular formula is carbon 5, hydrogen 10, oxygen 5. Then the aldose form is ribose sugar or xylose sugar, and ketose form is ribulose and xylulose form. Then comes to the 6 number of carbon atom. When the number of carbon atom is 6, the sugar is said to be as hexose sugar. Its molecular formula is carbon 6, H12, oxygen 6. Aldose form of hexose is glucose, galactose, mannose. You remember there are 3 form of aldose sugar of hexose, that is glucose, galactose, mannose. Similarly, the ketose form of hexose, that is fructose. Then comes to 7 carbon atom containing sugar, that is called as heptose. Molecular formula is carbon 7, H14, oxygen 7. Aldose form of heptose, that is glucoheptose and galactoheptose, and the ketose form of heptose, that is pseudoheptulose, that is called as pseudoheptulose. Okay, my dear student. Then comes to properties of monosaccharide. First one is the monosaccharide is colorless, crystalline, water soluble, sweet taste, and low molecular weight. Low molecular weight means its molecular weight is less than 100 Dalton. That is sweet taste. Then colorless, that is a crystalline, sweet, water soluble, sweet taste, and low molecular weight. All monosaccharides having asymmetric carbon atom. Asymmetric carbon atom means the carbon atom has 4 functional group. Similarly, except dihydroxyacetone has no asymmetric carbon atom. All monosaccharides having asymmetric carbon atom except dihydroxyacetone compound. Triose sugar, this is a triose sugar which has no asymmetric carbon atom. But according to the number of asymmetric carbon atom, the number of isomer can be get by formula, that is 2 to the power n. N is the number of asymmetric carbon atom. We know glucose is a hexose sugar which has a 4 asymmetric carbon atom. This is one, this is another, this is another, this is another. Here 4 asymmetric carbon atom present in glucose. So its formula is 2 to the power n. So 2 to the power 4 is equal to 16 isomer. So glucose has 16 isomer. Here glucose has a two form of structure. One is a L isomer form, another is a D isomer form. We know isomer. What do you mean by isomer? Isomer means those compounds having similar molecular formula but different structure is there. Here D, here this is L glucose, this is D glucose. Why it is considered as a D glucose? When the fourth asymmetric carbon atom having OH group is located at the right side and hydrogen atom is located at the left side. So this type of glucose or hexo sugar, that is called as dextrorotatory glucose. Similarly, L glucose means when the OH group is located on the left side of the asymmetric carbon atom and hydrogen atom is located at the right side of the asymmetric carbon atom, then this is called as levorotatory form of the glucose. So this is called as L glucose form, this is called as D glucose form. In most of the biological system, the D glucose is common occurrence in all cell and different living system. Next come to the then monosaccharides having open chain structure and ring structure. We know it has two type of structure. One is open chain structure, another is a ring structure. We have studied about the open chain structure. This is the open chain structure of glucose. One is L isomer form, another is a D isomer form of glucose. Then comes to the ring structure of the glucose. Here there are two types of structure of hexo sugar, that is one is a pyranose form, another is a furanose form. What is pyranose form? This is a six membered ring structure. Six membered ring structure means the ring oxygen goes by one position goes to the oxygen atom and a 5 position goes to the carbon atom. Here this is the first position of carbon, second position of carbon, third position of carbon, fourth position of carbon, this is fourth position of carbon, this is a fifth position of carbon, this is oxygen position. Here this is oxygen position. So this is a hexagonal structure. Here this is also 5 position, this is 6th position. Here the carbon atom goes to the 5 position of ring structure and 1 position goes to the oxygen atom. So this is a 6-membered ring structure. Pyranose is the 6-membered ring structure. 1 position goes to the oxygen atom and 5 position goes to the carbon atom. So this is called as pyranose form of the glucose. Similarly, furanose form of the glucose, this is the 5-membered ring structure of glucose, but here one position goes to the oxygen atom and 4 position goes to the carbon atom. This is a first position, this is second position, third position, fourth position and fifth and sixth position. Here 1, 2, 3, 4. 4 position goes to the carbon atom and 1 position of ring structure goes to the oxygen atom. So it is a pentagonal ring structure of glucose. So this type of form of ring structure, that is called as furanose form of the glucose. This is the this is the fructose. This is the structure of fructose. fructose has furanose form, glucose has pyranose form. But this is the glucose form, this is the fructose form. But now comes to the next structure of the all monosaccharides are reducing nature because they have free aldehyde and ketone group. So, all monosaccharides are reducing nature. Monosaccharides having anomers, that is alpha and beta form. First, then we go to the pyranose form, glucose. Here glucose and fructose are the isomer with each other. This is the ring form of glucose. But actually, glucose, one things you know, epimer form. What do you mean by epimer? We know glucose, galactose and mannose, they are the isomer with each other. In case of galactose, when the OH group is located on the above position and hydrogen is located on the below position, all structures are same with the glucose, that is called as galactose. You remember the epimeric position of fourth carbon atom in galactose is isomer with the glucose. Similarly, mannose in the second carbon atom, when OH is located on the above position and hydrogen is located at the below position, all other structure are similar with the glucose, that is the epimer of the mannose. This structure is called as mannose structure. Glucose is epimer with the galactose at its fourth position of carbon, but when glucose is epimer with the mannose at its second position of carbon atom, means here OH is located in the above position and hydrogen is located below the below position. Similarly, in case of galactose, at the fourth carbon atom position, OH group is located in the above position and hydrogen is located at the below position. So, glucose, galactose and mannose, they are isomeric with each other. They are called as epimeric with each other, means they are converted into one form to another form in presence of enzyme, epimerase enzyme. Okay. Then comes to the monosaccharides having anomers. Anomers means additional carbon atom, additional asymmetric carbon atom formed in the glucose or fructose, that is called as anomers. Anomers is also one type of isomer form of glucose and fructose that may be found in the form of alpha form and beta form. Here you can see the picture and the structure of the glucose and fructose. Here this is the first carbon atom, second carbon atom, third carbon, fourth, fifth, sixth carbon atom. Sixth carbon atom is present in the hexagonal form. This is pyranose form. When the H is located in the, it is alpha form of glucose. Alpha, only anomers can be considered at its first position of carbon atom. First position of carbon atom. When hydrogen is located at the above position, then it is called as alpha form. When hydrogen is located to the below position, that is beta form. You remember, here first position of carbon, this is, here also first position of carbon is this. But here when hydrogen remains at the above position, that is alpha form. When hydrogen remains at the below position, that is beta form. That is beta glucose, and that is beta glucose, this is alpha glucose. You remember basic difference between alpha and beta form of glucose. when hydrogen remains at the first position of carbon atom at the above position, that is the alpha form. When hydrogen remains below at the first position of carbon, that is called as beta form. Here alpha and beta form of glucose can be distinguished from each other in this manner. We know all D form of glucose commonly found in biological system. Similarly, you come to the fructose. Here the anomer is only can be find out to its the second position of sugar. Second position of sugar. If this is the first carbon atom, this is second carbon atom, third, fourth, fifth, sixth carbon atom. Suppose CH2OH is located at the above position, the anomer form is alpha. If the CH2OH is located below form, the anomer form is beta. That is beta fructose, that is alpha fructose. You can distinguish between alpha form and beta form. Here when CH2OH is located at the above position, that is alpha form. When it is located at the CH2OH is located is at the below form, that is beta form. Alpha and beta form of glucose. Then comes to glycosidic bond. We know glycoside bond develop between two sugar residue. After formation of glycosidic bond, the sugar can be considered as a sugar residue. Only glycosidic bond is formed between two sugar units. That is here with removal of a water molecule. With formation of a water molecule, here this is a glucose, this is a fructose. It has a OH group, this is OH group. They combine with each other. OH plus H, that give rise to water molecule. Here only oxygen can form glycosidic bond between two compound, that is glucose and fructose molecule. But actually you can see that here this is the alpha form of glucose. This is the first carbon atom, alpha form of glucose. Here this is the second carbon atom, this is also alpha form of fructose. Here the glycosidic bond is formed, is considered as alpha 1,2 linkage. Alpha 1,2 linkage. Alpha 1,2 glycosidic bond. Here this is a glucose, this is a fructose. But here this is alpha glucose, this is alpha fructose. So the linkage is formed between alpha 1,2 linkage or alpha 1,2 glycosidic bond. Then comes to disaccharide. When two sugar residues joined by glycosidic bond, that form disaccharide. It may be three types: maltose, lactose, sucrose. Here maltose, this is commonly found in malt cereal, so its name is malt sugar. Maltose on oxidation, that give rise to glucose and glucose in presence of enzyme maltase. Here you can see the picture. This is the alpha D glucose, that is also alpha D glucose. Here this is the first carbon atom, this is fourth carbon atom. This is alpha form because H is located at the above position. Here H is located at the above position. This is also alpha form of glucose. But the bond is formed between first position of carbon atom and fourth position of carbon atom. Here the first position is carbon, second, third and fourth position. Here second, third, fourth position. But here the glycosidic bond is formed between first position of carbon atom and fourth position of carbon atom between two sugar, that is alpha glucose sugar. So the bond is called as alpha 4 glycosidic linkage or glycosidic bond. Then another, then comes to lactose. Lactose is commonly found in milk, so that is called as milk sugar. Lactose on digestion in presence of enzyme lactase enzyme that form glucose and galactose. Here galactose and glucose. Here fourth carbon atom OH is located at the above position and a hydrogen at the below position. So this is the first position, this is fourth position. 1 4, this is also called as a beta galactose, this is also called as beta glucose. Here OH is located at the above position, here also OH is located at the above position. So these are two beta form of the galactose and glucose. Here the bond is 1 beta 1 4 glycosidic bond or glycosidic linkage. Then comes to sucrose. Sucrose is also called as cane sugar, that is commonly found in sugar cane. So sucrose on digestion in presence of enzyme sucrase that form glucose and fructose. Here alpha glucose, here beta fructose. Here first position, this is second position of carbon atom. Here H is located at the above position, this is the alpha form of glucose. CH2OH is located in the above position, CH2OH that is located in the above position, so this is called as beta fructose. This is called as beta fructose. Alpha glucose and beta fructose, they are combined by a glycosidic bond that is called as a alpha 1 to glycosidic bond. Alpha 1 to glycosidic bond. So this is all about your sucrose. Then comes to the oligosaccharide. Oligosaccharide means when 3 to 10 sugar residue joined with each other by glycosidic bond that form oligosaccharide. That may be trisaccharide, tetrasaccharide, pentasaccharide up to decasaccharide. Trisaccharide particular example when 3 sugar residue are joined by glycosidic linkage that form trisaccharide. That may be particular example is gentianose, raffinose, stachyose. There are 3 typical example of trisaccharide. In gentianose, 3 glucose molecules are combined by glycosidic bond that form gentianose. Similarly, in case of raffinose, galactose, 1 galactose, 1 glucose, 1 fructose, they combined by glycosidic bond that form raffinose. Similarly, stachyose, galactose, glucose, 2 galactose and 1 glucose molecule combined by glycosidic bond that form trisaccharide, that is stachyose. These are different types of oligosaccharide found in living system. Then comes to fourth type, that is polysaccharide. Generally, when more than 10 sugar residues joined by glycosidic bond that form polysaccharide. Polysaccharide are non-reducing, they have no free aldehyde and ketone group, they are insoluble in water, optically inactive, tasteless, high molecular weight, means more than 1000 Dalton. Its molecular weight is greater than 1000 Dalton. So they are the macromolecule of the cell or living system. So they having high molecular weight. Types of polysaccharide. There are 2 types of polysaccharide. One is homopolysaccharide, another is heteropolysaccharide. Homopolysaccharide means when the sugar residue are same. If the polysaccharide is made of only glucose unit, then it is called as homopolysaccharide. If the polysaccharide molecule is made of only fructose molecule, that is also called as homopolysaccharide. Means when the sugar residue are same, they are joined by several glycosidic bond that form homopolysaccharide. Particular example, that is starch, cellulose, glycogen, chitin, inulin. They are the typical example of a homopolysaccharide. Then comes to heteropolysaccharide. Heteropolysaccharide means when different sugar residue are joined by glycosidic linkage, that form heteropolysaccharide. Suppose glucose plus fructose, they combined with each other, that form heteropolysaccharide. Different sugar residue are joined, that form heteropolysaccharide. Like heparin, we know heparin anticoagulant, that is also heteropolysaccharide. Hyaluronic acid that is found in different connective tissue, that is made of different sugar residue. Similarly, keratin sulfate, keratin means it is a protein which present in the hair, but it is keratin sulfate. Keratin sulfate is considered as a heteropolysaccharide, that is made of different sugar residue, but keratin is made of protein, but keratin sulfate is a heteropolysaccharide. Similarly, chondroitin means they are the protein present in the cartilage, but here chondroitin sulfates, they are the heteropolysaccharide. Heteropolysaccharide, they are made of different sugar residue. Similarly, peptidoglycan, pectin. Peptidoglycan, you know, bacterial cell wall component, that is peptidoglycan, that is made of NAM and NAG. These are the, here the different structures are given. This is the structure of starch. We know starch is a storage carbohydrate of plant, that is made of two chain. One is a unbranched chain, another is a branched chain. Unbranched chain is called as a amylose, branched chain is called as amylopectin, that is a 20 to 30% amylose chain and a 70 to 80% amylopectin chain. Here these are made of different glucose unit by alpha 1,4 linkage. Similarly, amylopectin is more branch, about on 25th number of glucose residue or sugar residue, the bond is formed, that is the branch point is alpha 1,6 linkage. After 25 number of sugar residue, a branch point is originated, which is formed by a linkage, that is alpha 1,6 linkage. In case of amylopectin, we know starch is the test of starch is done by iodine test. Amylose provide black color in iodine, whereas amylopectin gives violet red color in iodine test. These are the different starch structure. Then comes to another is glycogen. Here is the structure of glycogen. Glycogen is a more branch structure. It is especially the reserve storage of animal body, especially liver and muscle cell. Like glucose amount when deposited, that converted into glycogen. It is a amylopectin structure, not amylose structure. Here it is more branch structure. 8 to 11, after 8 to 11 sugar residue, one branch point is originated, that formed by alpha 1,6 linkage. It is more branched than amylopectin in case of starch. So then comes to then amino acid. Then amino acid, Amino acid, we know amino acid is the monomer or micromolecule which present inside the living system. So amino acid is considered as a monomer of protein. Suppose we consider monomer is the polymer, but amino acid is considered as a monomer. Means many amino acids are joined with each other by several peptide bonds that form a polymer that is considered as a protein molecule. But here amino acids are considered as a alpha amino acid. Why it is considered as alpha amino acid? Because it has a alpha carbon atom through which different amino and carboxyl group is attached. So it is considered as a alpha amino acid. Amino acids are also substituted methane. We know the structure of methane, that is a hydrogen, 4 hydrogen atom is bonded with a carbon atom. This is methane. Similarly, amino acids are substituted by methane. We know here the structure of amino acid is given in the board, but here this is called as a alpha carbon atom. It has a 4 functional group. One is a carboxyl group or acidic group, then this is amino group or base group, and a hydrogen atom, then a side chain or alkali. R means alkali group, that may be methyl, ethyl, propyl, butyl, like so on. So there are 4 functional groups which remain attached with a carbon atom. So this carbon atom is called as alpha carbon atom. Then here carbon atom of amino acid is chirality, chiral carbon atom, because it is connected with a different functional group. So it is also called as chiral carbon atom or bonded with 4 functional group, that may be amino group, carboxyl group, alkyl group, and hydrogen atom. This is also called as a chirality. That amino acid also shows chirality. We know there are 20 amino acids which participate in the synthesis of protein. So these 20 amino acids are called as magic 20. This is also called as magic 20, or this is also called as standard amino acid. Means they can participate in the synthesis of protein molecule, so they are called as standard amino acid. And other amino acids are also available, they are called as a non-standard amino acid, non-protein amino acid. About 300 to 400 number that can form different type of protein. They are available about the 300 to 400 number of non-standard amino acids are also present. But actually when the alkyl group is a hydrogen atom, the amino acid is called as glycine amino acid. You remember, glycine is the simplest type of amino acid. When the alkyl group is a hydrogen atom, it can be considered as a glycine amino acid. About 20 amino acid, glycine is the most simplest type of amino acid when the alkyl group is a hydrogen atom. When the alkyl group is a methyl, the amino acid is considered as a alanine. When the alkyl group is a methyl hydroxyl CH2OH, the amino acid is considered as a serine amino acid. Serine amino acid is alcoholic amino acid. Okay. Then comes to amino acid is amphiteric compound. Why it is considered as amphiteric compound? Because it has a 2 functional group. It has a 2 acidic group as well as basic group or base group. It is a compound which possesses both acidic as well as base group. So it is called as amphiteric compound. Amino acid is deuter ion or dipolar ion. it forms dipolar ion. You can see the board here. The pH, the solution where the pH is neutral, neither positive or negative, here the carboxyl group is dissociate and amino group is protonated. Means carboxyl group is proton donor and amino group is proton acceptor. This type of electrical point, or this is a neither positive charge nor negative charge, so that point is called as a isoelectric point. This type of ion is also called as dipolar ion, or this is also called as Joiter ion. So amino acid is also called as a Joiter ion or dipolar ion. But at under alkaline medium, the carboxyl group is dissociate, whereas in acidic medium the amino group is protonated. Protonated means proton accept and that is a proton donor. Next comes to the classification of amino acid. On the basis of synthesis in animal body, it is again three types. One is essential amino acid, non-essential amino acid and semi-essential amino acid. What is essential amino acid? Those amino acids which are not synthesized in the human body. They come through the diet. This type of amino acid is called as essential amino acid. About 8 number of essential amino acid present. V for valine, I for isoleucine, P for phenylalanine, M for methionine, L for leucine, L for lysine, T for tryptophan, T for tyrosine. These are the 8 number of amino acid. They are considered as a essential amino acid which cannot synthesize in the human body. They only come through the special diet. Then non-essential amino acid, those amino acid which are synthesized in the human body, they are called a non-essential amino acid. And semi-essential amino acid, those amino acid which are partially synthesized in the human body and partially come through the diet, that is called as semi-essential amino acid. Especially arginine and histidine are semi-essential amino acid. Again, on the basis of amino and carboxyl group, it is again three types. One is acidic amino acid, basic amino acid, neutral amino acid. If the one more acidic group is present in the amino acid is called as acidic amino acid. That may be glutamic acid, aspartic acid. One more amino group is present, the amino acid is called as basic amino acid. That may be lysine and arginine. Neutral amino acid, neutral means equal number of acidic and a base group is present in those amino acid. That is called as neutral amino acid. That may be glycine and alanine. On the basis of nature of side chain, they may be classified into 4 groups. First one is amino acid with aliphatic hydrophobic side chain. That may be alkyl group contain aliphatic hydrophobic side chain. That may be glycine, alanine, valine, leucine, isoleucine, proline. Amino acid with aromatic hydrophobic side chain, that may be phenylalanine, tyrosine and tryptophan. Similarly, amino acid with sulfur containing side chain, that may be cysteine and methionine. Cysteine and methionine are typical sulfur containing amino acid. Then comes to amino acid with alcoholic group. If the side chain contain alcoholic group, the amino acid is called as alcoholic amino acid. That may be serine and threonine. Serine and threonine. Then comes to peptide bond. We know peptide bonds involves between the carboxyl group of first amino acid and amino group of second amino acid with removal of a water molecule. Here this is first amino acid, this is second amino acid. This is the carboxylic group of first amino acid, this is the amino group of second amino acid. Here the peptide bond is formed between the carboxylic group of first amino acid and amino group of second amino acid with removal of a water molecule. Here OH group is combined with a hydrogen atom that form water molecule. So only CHCO group is present, that is called as amide group. CHCO, that is amide group, that is also called as peptide bond. Here peptide bond, after formation of peptide bond, the amino acid is called as amino acid residue. Okay. One formula is the number of peptide bond is equal to A minus 1. A is the number of amino acid minus 1 when suppose 4 amino acids are present in a peptide. 4 minus 1 means there are 3 number of peptide bond found in a peptide. So this is the peptide bond. Peptide bond is responsible for synthesis of protein molecule or polypeptide. These are the last part of the amino acid. In the next class we will discuss about protein and lipid.